in our final lecture, we'll talk about nucleotide metabolism. So this is how we build our purines and pyrimidines from scratch, as opposed to getting them in our diet. And we also need to talk about how we convert nucleotides into deoxynucleotides for DNA. So I broke the lecture down into very simple parts, and they should have these colored headings at the top of each slide as you go through and review. So we're going to first talk about how we make our nucleotides, right? So the anabolism or synthesis of purines and pyrimidines. Then we'll convert to breaking them down, our purines and pyrimidines again, and so we can turn them into waste products or other materials, or actually have a little bit of a pathway in between where we scavenge these things to turn them into others. And at the very end, we'll talk about how we turn nucleotides, that's our RNA type molecules, into deoxynucleotides, the DNA molecules. So we need to remove that oxygen at carbon 2'. So let's start with our nucleotide synthesis from the beginning. And so I need to briefly remind you what a nucleotide is. So looking at the figure at the bottom, on the bottom left, we have the basic outline of all of our nucleotides, be they ribonucleotides or deoxyribonucleotides. So we have a pentose sugar, right, ribose, and it's either going to have one of our 5 bases on it, right, attached at carbon 1'. That's our new glycosidic bond. And then at the 2' and 3' positions, we have hydroxy groups for ribose. If it's deoxyribose, the 2' is just a hydrogen. And then a 4' is the oxygen is still in the ring because we formed its hemiacetal form. And then on the 5' carbon position, that oxygen is now occupied by one or more phosphates. If it's no phosphates, we call it a nucleoside. If it's got one or more phosphates, it's a nucleotide, but we name it nucleoside number of phosphates. So for instance, if you have all of the things shown here, it would be a nucleoside triphosphate, which is a nucleotide. Okay. So on the right are our 5 bases again, 2 purines, 3 pyrimidines, a subset of which is found in RNA and DNA, the difference being the thymine in DNA versus uracil in RNA, and the only difference in those two is the presence of that methyl group. Excuse me. So if you remember our structures of pyrimidines and purines, that each ring structure, whether it's a purine or a pyrimidine, has 2 nitrogens in it. So every ring has 2 nitrogens in it. Adenine has absolutely no oxygens. Guanine has 1. Cytosine has 1. And both uracil and thymine have 2. And if you remember how they pair together, adenine would pair with either uracil or thymine for a grand total in the pair of 2 oxygens. And guanine would pair with cytosine for, again, a grand total of 2 oxygens. So how are these molecules assembled? Well, let's focus on the rings themselves. And we'll start with the larger one, the purine on the left. So it starts here, not at number 1, that's just where it's labeled. We're going to start here at number 9, right? That's just its label, but that is the first atom assembled. So purine rings are going to be assembled in a different way from pyrimidines. So in a purine, you first build the sugar, the ribose, with the phosphate on it. And then on the ribose, you build the purine ring directly on the ribose sugar. Okay. Whereas with pyrimidine rings, you build the ring entirely separate. The 6-membered ring there for the pyrimidine, you build it entirely separately from the sugar, and then you attach it to the ribose. So it's a different way of making each one. Purines are made on the ribose itself. It's assembled stepwise on a substrate, on the ribose frame, whereas pyrimidines are assembled separately in solution and then attached to the sugar as a whole. Okay, so let's go through these first. We'll start here with the glutamine. Right, so glutamine puts the first nitrogen, it's going to be the donor, this nitrogen number 9 here, is the first place that we add something to the sugar. Right, and I'll go over this in the next slide. So we're going to put this nitrogen here on carbon 1 prime of the ribose. Then we're going to add these 3 atoms, the green ones here, from glycine, because these 2 carbons and the nitrogen constitute a glycine molecule, missing some parts, which we'll leave out here for a second. But we put that on this nitrogen next. Right? Then we add glutamine over here onto that carbon. Right? Then we close this ring, right? We close this first 5-membered ring with a carbon we get from tetrahydrofolate. Okay, that's our donor for this one. So far it's been amino acids and 1 carbon from tetrahydrofolate, one of your vitamins. Now you see why folic acid is so important early on. We need it to metabolize nucleotides, purines. Then we'll add this CO2 up here, right? And then we'll add add the carbon from CO2. We'll add another nitrogen from yet another amino acid, aspartate. And then lastly, we'll close this 6-membered ring with yet another carbon from tetrahydrofolate. That emphasizes its importance here. It's used twice to make every purine. Okay? And after that, the whole thing is already attached to a ribose, so it's completely finished. As opposed to pyrimidines, well, we'll start by combining a glutamine, a CO2, and an aspartate into a 6-membered ring. Right? There's only a few sources of these atoms here. These 4 come from aspartate alone, right? And then a CO2 donates a carbon, and a glutamine a nitrogen, and then this whole thing is then attached to a ribose at carbon 1'. So purines obviously have more donors because they're larger, and pyrimidines have fewer because there's fewer atoms. Okay. How do we make these from the start? Let's look at the left side here. We start out with a ribose 5-phosphate. Now where would we get something like ribose 5-phosphate to start? Give me a source of ribose 5-phosphate.
Glycolysis.
So glycolysis would never make a ribose. It only has 6 carbon sugars, or 3 carbon sugars. It doesn't have 5 carbon sugars. We can make it from glucose, if you remember. When we do the oxidative version of the pentose phosphate pathway, the oxidative phase, we can turn our glucose 6-phosphate into ribulose 5-phosphate. And ribose and ribulose 5-phosphates are just isomers of one another, much like glucose and fructose are isomers of one another. Okay, one's a ketone, one's an aldehyde. So this is readily available from sugar stores, right? Then what do we do to it next? It looks like we take our ribose 5-phosphate and add 2 more phosphates right here. Right, so ATP is donating 2 phosphates, leaving an AMP behind. So this costs me 2 ATP equivalents, right? This is my ATP debit card I'm charging twice. And both of those phosphates end up on carbon number 1. Right, so a pair of phosphates on carbon number 1. Sounds like these 2 would make a great leaving group in a minute. We've seen that game several times. Right? In the next step, we use glutamine to donate an amino group, leaving behind glutamate. Right? So water's cleaving off the last amino group of glutamine, leaving glutamate behind. And of course, phosphates are leaving. There is a mistake in this picture. This PI here should say what? What should this inorganic phosphate read instead?
As 2 PIs?
It should be pyrophosphate, exactly. So PPI, right? We've seen that many times before. It's a great leaving group. Now, they could have corrected this, just put a 2 in front of it, because the pyrophosphate, you know, in the next step would be hydrolyzed to 2 orthophosphates. So correct that in your figure. This should say pyrophosphate, PPI, which then in a subsequent step would be degraded. We've seen this many times in many pathways. It's a great leaving group. And so that ends up with a nitrogen attaching itself at carbon number 1 here. So there it is. Now I have my phosphoribosylamine. All right? So it's a phosphoribose with an amine on it. So phosphoribosylamine. Very simple. The enzymes that did this, right, there are 2 steps here. It's Prpp synthetase. Prpp stands for what? A phosphoribose pyrophosphate. It's already got 1 phosphate on a ribose, and I added 2 more. It's my ribose 5-phosphate plus 2 more phosphates. Clearly they labeled them all out here. So a phosphoribose pyrophosphate. Right? And then lastly, we want to transfer onto that an amino group. So it's PRPP amido transferase. So I'm taking the amino group off of here, right, putting it on carbon number 1. Carbon number 1 is losing a pyrophosphate as a great leaving group. Okay, we'll get to the unusual regulation a little bit later. Okay. So all this stuff just done here is take my ribose 5-phosphate and turn it into a ribose 5-phosphate with an amino group on number 1. And at this point, I can continue building this purine, right? Which can go back to the previous slide. That's this nitrogen right here from that glutamine. Right? Then I continue building this. I put glycine on next. Then I put this other amino group from glutamine. And I close the ring with this carbon from tetrahydrofolate. I add the CO2 up top, right? Then I add a nitrogen from aspartate. And then I close this ring with tetrahydrofolate. You notice both rings are closed by tetrahydrofolate. Okay? And once I have all those pieces on there, it's something called IMP. I being inosine. We've seen the structure of inosine once before, right? We were talking about how the RNA in tRNAs and mRNAs interact, but that's inosine. And in fact, inosine is a precursor for both of our purines, A and G. Right? So if you go back and look up the structure of inosine, you'll see that it's kind of a little bit of a hybrid between A and G. Okay? So what we have here is a phosphate on a ribose. I've built the rest of this purine ring out, like I showed on the previous slide. And at the point where I finish building it, it's called inosine monophosphate because it has one phosphate. At that point, we can go one of two ways. We can take the inosine, right, and turn it into an adenine, right? So inosine becomes adenine, right? Or the inosine becomes xanthine. And that's our branching pathway to make A's or G's. Okay, so when we form the adenine, we need to do something. Right? So we're going to hydrolyze a GTP to attach an aspartate. That's all we're doing, attaching an aspartate. What's the point of attaching an aspartate? We've seen this game on the previous slide. When did I use an aspartate here? Well, on the left, I've used an aspartate to give it a nitrogen. So a nitrogen donated from aspartate is an excellent way to do that. So I'm going to do it here. I use the power of GTP to put an aspartate on, right? Actually, I put a phosphate on first and the phosphate leaves, but that puts the aspartate's nitrogen on to the IMP. And if you go back and look at the structures of inosine and adenine, you notice that we need another nitrogen to make it an A. We need that fifth nitrogen. And that's exactly where it comes from, aspartate. Whereas with guanine, I need to turn the inosine into xanthine. That's simply turning an oxygen into a carbonyl, right? And then I need to turn glutamine into glutamate. Again, that's an amino donor, right? So I'm going to put the amino group on to turn it into a G. Okay, so I end up with inosine becoming adenine or inosine becoming xanthine to guanine, both in their monophosphate form. Okay? And then all I do from here is phosphorylate them. AMP becomes ADP becomes ATP. GMP similarly becomes GDP to GTP. Right? And that's just phosphorylation events. Okay, so here's that process one more time, showing you where the carbons and the nitrogens are coming from. So we started out here. I had my ribose and a phosphate, and this nitrogen was put on already. Remember, if we go back, the nitrogen came from glutamine, right? So that's why glutamine is already attached here, this nitrogen. And then I attached this glycine, the blue part. So back here again, glutamine here, and the green ones, unfortunately it's a blue thing here, but these three green atoms are these three here. Right? So the two carbons and the nitrogen are these three there. You notice this is attached to the nitrogen that came from glutamine. There it is. So I make this chain here now. Right? In the next step, we turn this amino group, right? Attached to this amino group, we put something else. We put one more carbon. There's my tetrahydrofolate carbon right there. Okay, so I attached the carbon from tetrahydrofolate. And also I said we attach this other nitrogen over here from glutamine, and that's this one over here. Right, so I've attached the oxygen here, well, carbon and oxygen here, and I've attached the amino group down here. Right? The next thing was to make this into a ring. So you see it cyclizes into a ring. We lose this oxygen in the process. And I have my first 5-membered ring of the 2-ringed purine. Okay. And the next step, I said we're going to add something to it. I need to add the carbon up here from CO2. You see it comes on as a carboxyl group. There's the carbon from that. And I also need to add the nitrogen from aspartate in much the same way that I did before. Okay, so here's the CO2 being put on, right there. Then from aspartate, here's the aspartate molecule, really big molecule sitting out here. I'm only going to add this nitrogen. Okay, it goes directly on the CO2, right? The carbon from the CO2, so directly on here. Here's the entire aspartate molecule, right? There's the nitrogen of it. I attach it, and then the rest of the molecule leaves. Aspartate leaving without its nitrogen would be what molecule? So this part right here, this is an aspartate. Everything I'm circling is aspartate. It's leaving its nitrogen behind. There it is. And all of this is going to leave. What is the part that leaves? What is it?
Succinic acid.
Say again?
Succinic acid.
It is. It's succinate or succinic acid, right? And shown in this form, it would be succinic acid, but succinate, right? So another molecule from our citric acid cycle is a byproduct here. Right? So this molecule leaves as succinate, and I'm left with my nitrogen behind. The only thing to do now is connect these 2 nitrogens, right? And the way to do that is I add my final carbon from tetrahydrofolate, right? So I connect these 2 nitrogens by a carbon from tetrahydrofolate, which is not shown in this picture, but you end up with the fused ring of a 6-membered ring. Right, so here's the carbon of that anyway, and then we connect these 2 atoms right here, and we get our 6-membered ring. Okay, once our fused ring here, there it is, the IMP, right, Inosine Monophosphate, I need to do one of 2 pathways. We said we could turn it into an A or a G. So here's how we do that. Turning it into an A is rather simple. We just need to add another nitrogen. Right? So we add another nitrogen once again in the form of aspartate. Right? And when aspartate leaves without its nitrogen, you would think it's leaving yet again as succinate. But because it's adding a nitrogen and because we need to rearrange some double bonds here, it gets a double bond in itself and leaves as fumarate. Okay, I don't need you to know the mechanism of that. We won't cover it for this class. But it leaves a lot like succinate did, just leaves as fumarate. And now this oxygen is gone, right? And I have my adenosine with 1 phosphate. Then adenylate kinase does what kinases do. It phosphorylates it to ADP. Where do you think adenylate kinase gets the additional phosphate it's adding to this one? Who's the phosphate donor? So I have an AMP, and I'm phosphorylating it to make it ADP. Where did adenylate kinase get the phosphate? It's an obvious right answer.
Just a floating phosphate?
Now, where do kinases generally get their phosphates? They're transferases.
From an ATP?
From an ATP, exactly. So an AMP that binds to this enzyme, this one here, and uses an ATP to make the ADP. What's the other product?
Another ADP?
Another ADP, exactly. Right? So an AMP plus 1 ATP turns into 2 ADPs. That's the role of this enzyme. And of course, our next enzyme might seem a little silly at first, and it truly is for this step, but we'll see why in a second. We need it. An ADP is acted on by another kinase. It's a nucleotide diphosphate kinase. So it's a kinase that works on nucleotides that already have 2 phosphates. So it gives them their third. So So nucleotide diphosphate kinase puts a phosphate on ADP to make ATP. From whom does it get the extra phosphate?
An ATP.
An ATP. So what's the other product?
ADP.
And we accomplished nothing, correct? We turned an ADP and an ATP into an ATP and an ADP. It actually accomplished absolutely nothing. But this enzyme will continually do that. It doesn't hurt anything to do that. It just doesn't accomplish anything. But this enzyme is also used for all the other nucleotide diphosphates, such as GDP. Now it's accomplishing something. Okay, so let's go through the GTP pathway, and I'll explain how it accomplishes that at the end. So starting with inosine again over here. In the case of inosine, I've got my oxygen that the guanine needs, no problem, right? But I need to add another amino group elsewhere. I don't want to remove this oxygen, but I need to add another amino group over here. So I need to reduce this double bond, right, to a single bond so I can add something, right? And then I need to add an amino group. The donor this time, not aspartate, is glutamine. We've seen glutamine donate nitrogens before. So glutamine donates the nitrogen and becomes glutamate. How does it get it to put it on right here? Well, we first put on a pyrophosphate. That should look familiar. And then the glutamine attacks that, puts its amino group on, and the pyrophosphate leaves. This is exactly how we got the first nitrogen on the ring of ribose. It also used a glutamine. So it's the exact same chemistry. So why reinvent the wheel when it already exists? So XMP, xanthine monophosphate, to the GMP is the exact same chemistry as putting this first nitrogen on the ribose ring at the very beginning. They work exactly the same way. They first put on phosphate, the pyrophosphate, sorry, and then the nitrogen of glutamine displaces it. Glutamate leaves along with pyrophosphate. So this is driven forward as well. Okay? And then, of course, we make GMP. There is a kinase called GMP kinase, like there was an adenylate kinase, and it puts the second phosphate on at the expense of a GTP. So GMP plus an ATP becomes a GDP and an ADP. That did accomplish something, right? And then finally, the nucleotide diphosphate kinase does something useful. It turns a GDP into a GTP, but of course at the expense of an ATP. ATP becomes ADP. This is why I keep saying GTPs and ATPs and CTPs and all those, they're all equivalent in terms of energy, right? We keep calling them, or I keep calling them, ATP equivalents because of this enzyme. A GTP and an ATP are interconvertible in the cell, as well as a CTP and TTP and UTP and all the others. Right? They all have the high energy phosphates, and this nucleotide diphosphate kinase is able to interconvert any of them. This one seems kind of silly that it does this at the bottom, but it works on any of them, including A's. But it makes sense that it will work on any of them. But so it is useful. It's just when it does an ADP to yet another ATP, we can laugh at it for accomplishing nothing. Okay. So how do we regulate these two pathways? How can we make an equal proportion or stoichiometric amounts of ATP and GTP? Here's how the pathway works. The same figure from the first part. I just put in all the regulation here. If I turn ribose 5-phosphate into the IMP, build the ring on it, turn it into put the nitrogen on, make it the adenylosuccinate, lose the fumarate, make AMP, phosphorylate it all the way to ATP, right? And I do the same other pathway over here, making GTP. Each of these, AMP and GMP, inhibit their own production. So AMP, if you make a lot of it here, goes back and inhibits the very step where it branched to the A side. Right? So AMP inhibits this step, turning IMP into adenylosuccinate. Similarly, GMP inhibits its own production. It prevents the conversion of inosine to xanthine. Right? That makes sense. It's fully inhibits their own production. But that doesn't mean you make these in stoichiometric amounts. It just means you don't make more than a certain amount of either one of them. They're not tied together just yet. But finally, AMP and GMP alone, either one of them, can slow down the production of IMP, the purines here, but it takes both of them to turn it off. Now they're intertwined. Let's say you always go down the GMP pathway. Let's just make that assumption for a minute. Let's see what would happen. It means you make a lot of GMP and almost no AMP. Well, the GMP is going to shut down its own production here, right? Because you just made a lot of it. But because I only have GMP made and not AMP in abundance, I don't really shut down the purine synthesis at the top. I'm still going to keep doing this. But because of the abundance of GMP, I'm kind of roadblocking this direction, and so it's going to be forced to go down the AMP pathway, thus making more AMP. And as the AMP builds up, it will come to a stoichiometric amount with the GMP, and together they will just shut down production. I could do the opposite scenario, where if I only make AMP, it will again inhibit its own production, but only partially inhibit formation of the purines, and IMP will be forced to turn into GMP. So I always get them in stoichiometric amounts because they inhibit their own production fully, but partially inhibit the production of IMP each. It takes both of them together to inhibit the synthesis of more purines. That's why they're linked together. So I'll always get stoichiometric amounts of AMP and GMP. And of course, ATP and GTP could be made from those. Okay. So moving on to pyrimidines, this one's actually much simpler because it's a smaller molecule. So let's start at the beginning. We're going to start with this glutamine. It's this red glutamine here, right? And it comes from glutamine. So we have a glutamine molecule. And to it, we're going to add some phosphates, right? So we add a phosphate onto it. We add a phosphate onto a bicarbonate. Here's my bicarbonate in the center of the screen. And those two together each can lose some phosphates. That's wonderful. And as they do, they bind together and make this new nitrogen-carbon bond. Right? So we have a nitrogen and a carbon hooked together. We put two phosphates on. We lost one of them. So I have my nitrogen, my CO2, and one phosphate left. That's called carbamoyl phosphate. Okay, so CO2 in the middle, you know where it came from? Bicarbonate. The nitrogen on the left here, labeled number 3, came from glutamine donating its nitrogen to become glutamate. And then ATP donated the phosphate here. The other one left as a leaving group, so I have 3 things still hooked together. Is carbamoyl phosphate a stable arrangement?
No.
No. For the last time, why not?
Beta keto.
It's beta keto one more time. It's the last time we'll do it this semester. But here's one more beta keto. That is a very common theme we've seen a number of times. So what would likely happen to resolve this? How do you solve this puzzle? It's unstable. How do we make it stable? We could do one of 2 things. Go ahead.
The phosphate leave.
The phosphate could leave, and that's exactly what we're going to do, right? Or we could do what to this carbonyl? We've also seen this in other pathways. We just saw it.
Or reduce it?
Sorry, say again?
Reduce the carbonyl?
You could reduce it to a secondary alcohol, correct. But that's less likely here because of the nitrogen next door, right? It's very difficult to reduce that to an alcohol when you have this amide type linkage next to a phosphoester, and it's much more likely just to lose the phosphate. So that's exactly what we're going to do. We'll lose the phosphate, okay? And as we lose the phosphate, that's going to leave a molecule behind, right, that is very likely to attack an aspartate or be attacked by aspartate. So here's my aspartate coming in. That's the rest of this structure. So we've already accounted for in 2 steps all 6 members of the ring. So here's my aspartate, all 4 carbons. If I lose this phosphate, I'll have a carbon and a nitrogen here. The amino group here is going to attack this carbonyl because the phosphate just left. Right? Phosphate left, amino group and carbonyl are going to bond together, so I get this carbamoyl aspartate. Is carbamoyl aspartate a stable arrangement? Do you see any beta ketos in there?
No.
No beta ketos. But I do have something that would love to leave, and I don't want it to happen. Right? We're not going to cover this lecture in this course. We would if we were doing the full metabolism course. But if you look right here, right, what is this molecule? Right? It's labeled 1, 2, and 3 here, not 4, 5, and 6. But in the carbamoyl aspartate, the product of step 2, the atoms labeled 1, 2, 3, nitrogen, carbon, nitrogen. What is that if it were to leave? The carbonyl with the 2 nitrogens on it. If it were to leave as that, what would that product be? It's a very stable waste molecule.
Asparagine?
No, not asparagine, just these 3 atoms, or these 3
Right.
nitrogen, carbon, nitrogen. The numbers labeled 1, 2, and 3. So it's a carbonyl with 2 amino groups on it. What would that be? I know we haven't covered this lecture and we won't, but it's urea. Right? So a carbonyl with 2 nitrogens attached is urea. Right? So we could have a urea waste product here. And we'll see eventually that we do get a urea waste product when we degrade nucleotides, but we're building them here, and I don't want that to leave. So I immediately have the nitrogen labeled 3 here, attack the carbonyl labeled 4, and it forms the ring of 6. So in the picture up here, the one labeled 3, which is from our glutamate, attacks number 4 to create this new bond. So this is my new connection, the blue arrow. Right? So once I form those into a ring, everybody's involved in the ring except this one little carbon at the bottom, this carboxyl group at the bottom. If I only could get rid of it, I'd have my ring. Right? But I have this carboxyl group. So I make this molecule called dihydroerotitate. Right? And it looks fairly stable. I have a problem though, is I've got a CH2, right, in the middle of it, which CH2s are not found in nucleic acids. Right? So I need to do something to it. So if I remove one of these protons and electrons that go with it, and the other proton by Q, who's very good at that, you guys know this already. So I can take carbons 5 and 6 to each lose a proton and a pair of electrons between them, and I make a double bond between it. And it leaves as QH2. So that's called Dihydroerotitate Dehydrogenase, because it's a redox reaction. And that makes erotitate. Right? And erotitate is finally arrived at my ring. I have a pyrimidine. I have a single ring with 2 nitrogens in it, no CH2s. I do have that pesky carboxyl group still attached. Right? So I have this erotitate. But is erotitate a stable arrangement? The product of step 4 there, tell me if erotitate is stable.
Would that be considered alpha keto?
That would be.
That was a conjugated pi system too.
You're exactly right in both cases. So it's considered alpha keto because it's an alpha type arrangement. It's also conjugated to another double bond. So this is very stable. In fact, this is exceedingly stable. The very next step is I take this very stable pyrimidine ring. Remember, we make the pyrimidine ring first, and then we attach it to the sugar. So the next step, I take my phosphoribose pyrophosphate, right, the PRPP, the same one I made before, and this time lose the pyrophosphate on number 1, and in its place I attach this nitrogen. Right, so now this pyrimidine ring, this erotitate, is attached to my monophosphate. Okay? So it's now called, instead of erotitate, it's erotinidine because it's now attached to its sugar, much like adenine becomes adenosine, right, or uracil becomes uridine. So we have erotitate becoming erotinidine, and I have erotinidine monophosphate, OMP. And OMP is very stable. However, this carboxyl group is a problem. It's in the way. I need it out of the way so I can base pair. So I need to decarboxylate it. And you told me this was exceedingly stable. So to lose the CO2 is not easy. Right? The enzyme that does this is called OMP decarboxylase. And if you're in the lab course, you'll be talking about this in one of your assignments for one of our papers. But it's one of those enzymes we talked about back in our enzyme lecture, where without the presence of the enzyme, the chance of this decarboxylating is about once every 78 million years. Right? It's that stable. But in the presence of the enzyme, OMP decarboxylase, it does it every few seconds. Right? So that's a huge improvement. It's about 10 to the 17th fold improvement. It's crazy to think about how efficient that enzyme is. Right? And it has to do with how it positions everything, including the phosphates and all the other parts to make this happen. But once we finally lose the CO2, I arrive at UMP. This is uracil now. So if I lose the carboxyl group, I have uracil. So the first pyrimidine made is uracil. Of course, OMP is also a pyrimidine, but the first of our four we're used to thinking about is uracil being made. Right? So let's review one more time where they come from. We take the glutamine and the CO2 and put them together. Right? We need some ATP to assemble that, and we end up with a carbamoyl phosphate, which is not stable. It loses its phosphate, and at the same time we make a new connection between the carbamoyl group and an aspartate, right, to make a carbamoyl aspartate, right, which is fairly stable. But I don't want to lose the side group as urea, so I need to cyclize it. So an enzyme, the dihydroerotitate, cyclizes it, right? So we lose the hydroxy group on the carbon here, and then we make this dihydroerotitate. This is not very favorable. I need to put a double bond here to make it favorable, and that would conjugate all of these. So I reduce this using Q, and I get this erotitate, a very favorable molecule, a first pyrimidine. I then take my assembled pyrimidine, put it on the ribose with its 3 phosphates, losing the pair on number 1, just like we did with the beginning of purine synthesis, and I get my OMP. OMP is a very, very stable molecule, so it takes some effort to remove this CO2. OMP decarboxylase accomplishes that to make UMP. Okay, so that's how we arrive at UMP. It's assembled backwards of a purine. Purines are built on the sugar. Pyrimidines are built first, then attached to the sugar. Okay, so I have my carbamoyl phosphate. Remember which reaction that was? That was the very beginning one. It was very unstable beta keto, losing its phosphate and attaching itself to aspartate to make carbamoyl aspartate. That was this reaction right here, number 2. This is the key step here, right, to making pyrimidines. If I don't want to make pyrimidines, I don't do this step. We use carbamoyl phosphate for other things, but I don't do step 2 unless I plan on making pyrimidines. So this step clearly should be regulated, and it is. If you continue this process and make your UMP, ultimately all the other pyrimidines are made from uracil. We'll talk about that in just a minute, how we get all the others. But the cytidines and the thymines are all made from uracil, or from UMP. So eventually UMP gets converted to CMP. Right? We'll talk about that after many steps. And CMP is converted to CTP and finally inhibits the starting process. So we make sure we make enough U's so that we can make enough T's and C's as well. Now, only when we finally get enough C's, which is the end of the line, we inhibit the production of pyrimidines. Therefore, we always make enough U's, C's, and eventually T's. So just like purines, we can make enough of our products until they turn themselves off, turn the production of themselves off. But now I have a problem. I can make G's and A's in stoichiometric amounts, and I can make U's, C's, and T's in the right amounts until they inhibit their own production. But I have no connection between purine synthesis and pyrimidine synthesis yet. I haven't discussed any yet. And here's the connection. ATP will activate this enzyme. It will override the inhibition of CTP. So here's my connection between purines and pyrimidines. The purines tell the pyrimidines what to do. So if I make a lot of A's and G's, and I have yet to make an abundance of U's, C's, and T's, not so much T, but U and C, right? I have yet to keep up with the purines, then this concentration of CTP will be far less than ATP. And thus the ATP will override the inhibition and will make more pyrimidines. If the pyrimidines get more abundant than the purines, right, then the CTP will outcompete the ATP and shut this enzyme down. So that our pyrimidines aren't made until the purines are caught up to the concentration. So I have a connection here because this enzyme is sensitive to both ATP and CTP, right? It's ATCase, that should help you remember it's A and CTP that regulate this thing, right? ATP says make more, keep up with the purines. CTP says no, we have enough pyrimidines, don't make any more, right? Let the purines catch up. Right? So this enzyme is the linchpin that lets me connect purine and pyrimidine synthesis. And that's at the very beginning of pyrimidine synthesis. Okay? All right, the enzyme I was talking about, this very difficult enzyme, this Adenine monophosphate decarboxylase. It takes this very stable conjugated arrangement and decarboxylates it to a uracil. Okay? It's one of the most efficient enzymes known. I said it speeds up the reaction by a factor of 10 to the 17th. That doesn't mean the reaction goes faster. Remember what we mean by that. It means it happens 10 to the 17th times more frequently than without the enzyme present. So without the enzyme present, it's about once every 78 million years. With the enzyme present, it's once every few seconds, right? And the amazing thing about this enzyme is it uses absolutely no cofactors and no coenzymes. It does it all by the amino acids themselves, right? Which is fairly amazing. The amino acids alone can accomplish this chemistry. If you want to know how it works, read the paper that we're doing for lab. All right, so let's put these together one more time. So I have pyrimidine synthesis, CO2 and glutamine providing the first nitrogen and carbon. We make that carbon wall phosphate. Here it is, right? And then we regulate the very next enzyme, the aspartate transcarbamylase, the one that takes the phosphate off of this and attaches it to an aspartate. We said Ctp, which is made from the UMP eventually, comes back and shuts this down. This should be not an arrow, I know it's red, but it should be like a red T. It's an inhibition. UMP will also come back if you make enough of that and inhibit the first step of synthesis. So this is an auto shutting it down just like we had with ATP or AMP and GMP. But CTP, which is a farther downstream effect, shuts down the second step. Okay, how do we make CTP from UTP? Well, we make UMP into UTP. This is very unusual. I first put all the phosphates on, right? We didn't do that with the others. I put all three phosphates on, and then how do we get from U to C? The only difference being what? From uracil to cytidine. What's the difference? Look back at your structures. It's a common theme for today's lectures.
Methyl group?
I have to transfer not a methyl group, or methyl group's on the TTP. I'm going from U to C. Uracil to cytosine.
You need another nitrogen?
I need an extra nitrogen, right? I need another amino group, exactly. So I'm also going to lose a water molecule along the way, but I need an amino group. And the amino donors, that's kind of the theme for today, are going to be your amino acids. Glutamine, aspartate, glutamate, those are all your donors for amino groups. So glutamine is going to donate us an amino group like it's done several times already in this two pathways and become glutamate, making uracil into, or UTP into CTP. Okay? The only one left to describe is how we get to TTP, and I'm saving that for last. Okay? So here's my UMP one more time. I turn it into UDP and then UTP. This should look familiar. We have a kinase that works on U's and C's. We had one that was specific for AMP. We had one that was specific for GMP. And now we have one that works on U's and C's. It can't tell them apart. It works on both of them. All right, so we have a uracil cytidine monophosphate kinase. And of course, just like the others, where does it get its phosphate? From ATP. So a UMP becomes a UDP at the expense of an ATP. And then our friend, the nucleotide diphosphate kinase, one more time, is converting UDP to UTP, again at the expense of an ATP. Once we get to UTP, however, and only when it has all three phosphates, this next enzyme will not work otherwise, we have our Ctp synthetase, right? Where it takes the oxygen off, it turns it into a water molecule, and puts an amino group on, just like you just told me, at the expense of a glutamine. Glutamine's the donor, becomes glutamate. Glutamate is getting this oxygen ultimately, but could go through a water intermediate. And that's just like we saw with our amino acid synthesis in the last lecture. We turn one amino group into a carbonyl, and we turn another carbonyl into an amino group. This is exactly like that reaction. So Ctp synthetase behaves just like our other enzymes that transfer an amino, or do the amino for carbonyl swap. That's exactly what we're doing. Okay? All right, so once we've made CTP, this thing can go back and inhibit its own synthesis, as we showed on the previous slide. Right? And this is also balanced by ATP inhibiting the same reaction. And so if there's more ATP than CTP, we'll make more pyrimidines to catch up. If there's more CTP than ATP, we'll make more purines to catch up. So purines and pyrimidines are now balanced in their synthesis. Right, so let's move on to how we degrade these things since we just built them. So let's talk rather briefly about how we degrade them because we only make a few products here. So if you have RNA or DNA, generally this is RNA or DNA you ingest in your food, right? So unless, well, even then, I was going to say unless you're licking rocks, you're getting RNA or DNA, but there's probably bacteria on those rocks, so you're still getting nucleic acids in your diet, right? So you can't avoid them in your diet, and we don't need to make that many of them because we recycle them. But our cells do divide and we do need more, and we talked about the de novo synthesis. But what do we do with excess? We need to get rid of them. Let's degrade all these things. Let's throw them away. We don't want to throw the whole molecule away because there's some useful parts on it. Let's recycle some of it. So we take our RNA and DNA and we use ribonucleases or just nucleases. Those are the ones for DNA, right? So nucleases or ribonucleases are deoxyribonucleases for DNA, and it chops it up into the individual nucleotides. So you're going to get a ton of AMPs, GMPs, CMPs, UMPs, and TMPs. Right? So we're going to chop them all up. Let's do the RNA version of that first. So we get Gs, Cs, Us, and As, monophosphates. Right? So they all have ribose on it. They all have one phosphate left. Because remember, your RNA and DNA only has one phosphate in the backbone between the sugars. So we cleave them all apart and end up with one phosphate on the 5' end. And of course we have our base out here. So let's do G first. We end up with a GMP. We immediately remove the phosphate, so it's a purine nucleotidase, because a nucleotidase would turn it into a nucleoside. It doesn't show it here, but the phosphate's the byproduct here. And then we need to take the base off the sugar. This should make sense. It's a simple disassembly. So we take our RNA, chew it up into its 4 components. We cut the phosphate off. Then we cut the sugar off. So when we cut the sugar off, you're going to use a phosphorylase. You've seen one of these before. When you use a phosphorylase to break this bond, what class of enzyme is that first?
Hydrolase?
It's a hydrolase, but it does not use water. What does it use as the saw?
Phosphorus?
It uses phosphate, right? It has a phosphorus in it, yes, but it uses phosphate, inorganic phosphate floating around. There's lots of them. In fact, we just cut one off, right? So this is very similar to another enzyme we've seen that cuts something off of a sugar using a phosphate. In fact, it's at the same position, number 1. And that was our glycogen phosphorylase. It cut off one glucose molecule from the rest of the chain. Right? So here's yet again an enzyme, phosphorylase, cutting something off the end of a sugar, right, using a phosphate. So very, very similar chemistry. So we end up with a guanine released, but not a ribose 1-phosphate, okay, because it was a phosphorylase. We end up with a phosphate on the ribose. So that's better than just using water because I have a phosphate on my ribose already, and you can see, you could probably bet the name of the enzyme. It's going to move this ribose's phosphate from number 1 over to number 5, and I can use it for other things. I can build another nucleotide if I want. I can use it in photosynthesis. So this ribose 1-phosphate is not waste. Let's keep it. Okay. For the ATP pathway, or the AMP part of it, so we have RNA again. We degrade it into its 4 components. One of those components is the AMP molecule instead of GMP this time. And we do similar things here, but we can do them in either order this time. Okay, so for the GMP, we cut the phosphate off first. Then we cut the sugar off for the base. Here we can do either thing first. So let's do it in the similar order. Let's cut the phosphate off, and you end up with adenosine, right? No more phosphate. And then we cut the sugar off of the base, again using a phosphorylase. Okay, so again we end up with the cut off piece, right? And ribose 1-phosphate. It's not exactly adenine getting cut off. It turns into hypoxanthine. You'll see why in a second. Okay, but we cut this off after we remove the amino group. So doing it this way, following the arrows, so we go across, down, and then finish. We cut off the phosphate, we remove the oxygen, we remove the amino group, we deaminate it, right? Remove the amino group using water, puts oxygen on, and then we cut it off of the sugar, again with a phosphorylase. Once again, I end up with ribose 1-phosphate. I can recycle that. And instead of having adenine, I have hypoxanthine. Hypoxanthine is the base of inosine. I've been calling it inosine. Inosine is the base with the sugar. So to give you a quick SAT type question, adenine is to adenosine as hypoxanthine is to inosine. I'll say that again. So adenine, just the base, is to adenosine, where it's on the ribose, is the same as hypoxanthine, just the base, as it is to inosine when it's on the sugar. Okay? They just have a different name for it. Okay? That's one pathway. The other pathway is to do the first two reactions in the opposite order. Let's remove the amino group first, right? Let's remove that amino group and put the oxygen back on. That's a deaminase again, right? Instead of adenosine deaminase, it's an AMP deaminase, a different enzyme, but accomplishes the same chemistry, right? So now I have IMP, right? This is inosine again. This is kind of where we started, right? I could turn this back into AMP or GMP if I wanted to. So if you're in your diet, you have a lot of excess AMP, I could probably turn some of it into GMP if I wanted to. All right? And the last thing again is to cut it off of cut the phosphate off and then cut it off of the sugar, and I end up with the exact same products. So you can follow two pathways here, right, to get to our conclusion. But just like GMP, I end up with a ribose still having a phosphate. I did cut it off, but I used the phosphorylase to put one back on. And I end up with either guanine or hypoxanthine as my two bases that are cut off. Okay? So either guanine or hypoxanthine is how I arrived here. We're going to recycle the ribose 1-phosphate, but what do I do with these bases if I want to get rid of them? Well, I need to turn them both into xanthine. Hypoxanthine is almost xanthine. What's the difference between the two? What's the only thing I need to do here between hypoxanthine and xanthine? The hypo gives it away. The prefix hypo means what in organic or in chemistry?
Too little.
It means missing one or less, right? So if you have hypochlorite, it's missing an oxygen. If you have chlorate, it has its full set of oxygens. If you have hyperchlorate, it has an extra oxygen. Hypo means one less, hyper means one more. So hypoxanthine is xanthine missing an oxygen. So xanthine oxidase, right, puts it on. Right? This enzyme you think should be called hypoxanthine oxidase, but it's the same enzyme we're going to use twice, so it does both. It got this similar name. They discovered this reaction first. So hypoxanthine is oxygenated, becomes xanthine, and guanine is almost xanthine. We just need to do our favorite reaction of the last two lectures, exchange an amino group for a carbonyl. Right? This is again one of our swaps. We turn an amino group into a carbonyl, and the donating molecule gets the amino group, and that's called guanase by the enzyme, both of which now arrive at xanthine. Then xanthine oxidase takes my xanthine and oxidizes it one more time, all the way on the end over here, and makes what's called uric acid. Right? And uric acid is then excreted in the urine and in feces. Okay? The enzyme xanthine oxidase is inhibited by a molecule called allopurinol. Right? Here's a picture of allopurinol. It looks a lot like xanthine. The only difference is the nitrogens are now adjacent to one another rather than spaced by a carbon. So because of that, the carbon and nitrogen in different places, as you see here, I don't have a position to put this carbonyl. Right? If these nitrogens are swapped, I can't put a carbonyl on here. So it inhibits the enzyme. It binds perfectly well, but it can't do the chemistry. So it's actually a very good inhibitor of xanthine oxidase. Why would you do that? Because if you have too much uric acid being produced, that's called gout. Right? So too much uric acid and it tends to fall out of solution or precipitate in joints, most likely in your ankles and knees and hips, right, and larger joints under a lot of stress. And the uric acid will form uric acid crystals, and that makes it very painful to move those joints. So sometimes you're given allopurinol to inhibit the formation of uric acid. But you're also going to get excess concentrations of hypoxanthine and guanine at that point. You really can't limit your nucleic acid intake in your diet. Like I said, if you're licking rocks, you're still getting nucleic acids. So we just have to prevent the formation of the uric acid crystals. Okay. Right, so let's finish off the salvage pathway here. Instead of degrading it all completely to urea and uric acid and things like that, let's take the adenine that we just got out of that, the guanine and the hypoxanthine, those are our three purine degradation products, and turn them back into other purines. So adenine is very simple. We put it back on the sugar. Remember, in the de novo synthesis, we built the adenine ring structure on the sugar. In our case, we have a free adenine floating around. We reattach it to the sugar. That's a different enzyme. It's just a transferase. And of course, as we reattach the adenine, we lose the same pyrophosphate once again and make AMP. And you know what happens from there. The adenylate kinase, right, puts one phosphate on, and then our favorite purine, not purine, but our favorite nucleotide, diphosphate kinase, puts the second one on, which is a silly reaction to do, at least for ADP. The guanine, again, with a similar enzyme, gets put back on the sugar, very same pathway. And then the hypoxanthine, with the exact same enzyme, put the guanine on, puts the hypoxanthine back on to make inosine. This enzyme does either one. You just can't do adenine. It has a specific enzyme for adenine. But once we make IMP, you already know this pathway. We can turn it into A or G, right? And we can make ATP or GTP. So because of this salvage pathway, if I have in my diet too much purines, I can just degrade them all. If I have plenty of one purine and not enough of another, I can convert one to the other. Right? I can convert guanine into adenine and adenine into guanine if I need to. So that takes care of our purines side of things. What about the pyrimidine side of things? Well, I kind of put it all in one figure here. And I have some colored arrows to help you understand what's going on. If you move in this figure in the up and down direction by that amber arrow that says exchange, then you're exchanging between the U side or U's to C side at the bottom. So uracils at the top and cytosines at the bottom. Okay? You're exchanging one to the other. And exchange from U to C is just, or C to U, is the addition or removal of an amino group, right? So from U to C, we just need to add an amino group and lose a water molecule. And from C's to U's, we lose that water and add an amino group, right? Very simple. Right, so if you go to the left on this pathway, the red arrow, it's the degradation pathway, like we did with all the other purines. If you go to the right, it's like our last slide, how we salvage it. Maybe I have plenty of C's and not enough U's. I can convert C's to U's. Maybe I have plenty of U's and not enough C's. I can convert U's to C's. So using the purple direction to the right, salvage, along with exchange, I can recover the ones I need from the ones I have. And if I have too much, I can use the red arrow and break it down completely. So let's go through these steps really quick. I'm going to start in the very center of the diagram where it says UMP and CMP, because those are what we would get if we degrade nucleic acids, or RNA. Okay, so from here, from UMP, I can do something really simple. I can phosphorylate it to UDP, that's reaction number 6. I can also phosphorylate that to UTP, that's reaction number 3. So simply going from UMP to UDP to UTP. That takes up a quarter of this page with something trivial. Okay, so it's not that complicated. I can do a similar thing with CMP, right in the center of the screen. I can phosphorylate it to CDP. That's again reaction 6. It's the exact same enzyme. Right? We talked about that one already. Right? And then I can take CDP and phosphorylate it to CTP. That's reaction 3. Once again, the exact same enzyme. So half of my figure is something that you already knew. I can phosphorylate UMP or CMP to their diphosphate analogs, or their diphosphate analogs into the triphosphate analogs with my dinucleotide kinase. dinucleotide phosphate kinase. So now I have UTP and CTP. I know I can exchange UTPs at the very top right of the figure into CTPs by adding an amino group. We just did that early on. That's how we make Cs in the first place, right? And glutamine is my donor. So that takes care of reaction number 1 on the drawing. I can turn CTPs back into CMPs, that's reaction 4 at the bottom, just by cutting off 2 phosphates. So just use water to cut off a pair of phosphates, right? In fact, 2 waters, cut them off one at a time if you want. Right, so that's a very simple, and I've accomplished everything with this diagram to the right of the middle of the screen. UTP can be done the same way. I can cut off the 2 phosphates and get to 2 phosphates and UMP. So the entire right-hand side of the diagram is very simple, adding and removing of phosphates or converting of U's to C's, which we've already seen by reaction 1. The new reactions here are something we just talked about. I can turn CMP into cytidine. That's kind of like we did for purines. That's reaction 7 here. I'm just cutting off the phosphate. Very simple hydrolysis. Or I can use the UMP and turn it into uridine. Reaction 7 at the top. Again, just cutting off the phosphate. When they're in their monophosphate stage, I can't do anything with them. When they're in their triphosphate stage, I can convert U's to C's. That's what that enzyme does. It doesn't go backwards. And when they're in their no phosphate stage, the cytidine and the uridine, I can convert C's back to U's by reaction 9. So the enzymes that convert U's to C's or C's to U's are very specific about how many phosphates they use. U to C must have a triphosphate. C to U must have no phosphates. It only works on the nucleoside. Okay? And of course that leaves me with just one reaction left, and that's converting uridine here, right, into uracil. Right? It's the only one that gets cut off of the sugar. And it's just a hydrolase, and it releases it as ribose. Notice that's different. Right? In the purines, when I cut them off of their sugars, I used a phosphorylase, and it ended up with a ribose 1-phosphate. Pyrimidines, whichever one it might be, whether it's U's or C's, and of course T's works the same way. Whenever I cut them off, I just use a regular old hydrolase, and I get plain old ribose, no phosphate. That's a big difference. And of course I get my uracil. So all the C's, and effectively T's as well, and U's get turned into uracil and ribose. Now the only reaction left on the slide that didn't discuss directly is turning uracil in the salvage pathway back into these guys. But that's exactly like the de novo synthesis. We make the molecule first, then we attach it to the sugar. So that's reaction 12. Right? So we take the uracil, we attach it to PRPP to make UMP. Right? We could also take the uracil and attach it to plain old ribose and make uridine. Right? Where we lose the 1 phosphate from ribose 1-phosphate when we do that. Right? So either way, I can go back and forth from uracil to any of these things on this slide. Right? Just much like I can go from AMPs and IMPs and GMPs to any of the others. The only thing left to discuss in this whole pathway is how do we make T's, right, the thymine component, and how do we make DNA, because this has all been RNA so far. Okay? So here's how we make the thymine part of it. Right? So starting with uracils here at the bottom, so very bottom left of the screen we have uracils. Right? We can convert uracil into a dihydrouracil. Right? So it's right next to it, a uracil to dihydrouracil. What have I done? I've reduced that double bond. Now I have a pair of CH2s in it that's not normally found in our nucleic acid bases. Right? Then I break the ring with a water molecule. I open up the ring. I break this amide bond, right, between the two to give me a straight chain form now. Right? So I clearly break the bond between this nitrogen and this carboxyl group over here. Right? So I'm breaking this bond right there. Right? That gives me my 3-uridopropanoate. Urido is the word you've seen before. It was in our biotin ring, our urido ring of biotin, which just means there's a pair of nitrogens that are spaced with a carbonyl between them. Kind of looks like urea, right? And that's exactly what we're going to do. We're going to add a molecule of water, right? And a beta alanine leaves. Alanine is a carboxyl group, an amino group, and a methyl group, and a proton, or hydrogen, all on the same carbon. That's an alpha amino acid. We learned our amino acids were all alpha amino acids. Well, here's one that's in a beta amino acid. Here, the amino group is on the second carbon instead of the first. That's the only difference. See, the amino group here is attached to carbon, technically carbon 3, but carbon 2 of this chain, right? Instead of being at the alpha carbon, it's on the beta carbon, so carbon 3 instead of 2 of the whole chain. So water molecule comes in, cleaves off this side chain here. We get an ammonia molecule and CO2, right? We don't get in a urea directly, this nitrogen stays, but the ammonia and CO2 eventually become urea. We're not going to have a lecture on urea synthesis. We don't have time for that. But that's how we degrade our uracil into a waste product. Remember our purines, the waste products were a ribose 1-phosphate, which I could recycle, and a uric acid. Here, our pyrimidines, the waste products are just plain old ribose, no phosphate, which I could also recycle, and it turns it into urea. Okay. Thymine is much the same way, right? So thymine or uracil, basically with an extra methyl group, works exactly the same way. We reduce it, we put those carbons down to a single bond here, we add water once again, we cleave the exact same bond. The only difference now is we have a methyl group down here, right? So it's an isobutyrate this time instead of a propionate. And we cut it off, we don't get beta alanine, we get this amino isobutyrate, right? Which the isobutyrate can then be rearranged to convert into other amino acids. But again, we get our piece cut off, the very end cut off as ammonia and a CO2, which again are combined to make urea. Right? And then the last thing I want to talk about for the lecture is how do we make DNA? Right, so how do we make this molecule of ribose into deoxyribose? So for example, I want to turn a CDP into a deoxy CDP. I'm losing the OH on the 2' position. Okay? The enzyme that does this is called ribonucleotide reductase because this is a reduction. I have an alcohol and then I don't. Right? There's no OH here anymore. So it is a reduction. And it's a very, very, it's an interesting enzyme. Right? If we talked about last time how we discovered how an enzyme has allosteric regulation, right? The first one discovered. This one is the ultimate allosteric regulation, right? It's a programmable computer, if you think of it that way. It's a programmable enzyme. It has so much regulation and control over this thing. It's amazing how it works. And so what it essentially does, if you want the short version, is it takes a nucleoside diphosphate. It's got to have 2 phosphates, exactly. The base here, it could be almost any of them, not every one of them, but most of them. In this case, it's a C. We could use A's and G's as well and use. But we take a nucleoside diphosphate and turn it into its deoxy form. It will only do this if the base is already on there. It won't just grab a ribose and do this. So it's not like this is built on a ribose without its OH. Don't think that. It's built as a piece of, or a potential piece of an RNA molecule, and then turned into the deoxy form. Okay, so the nucleoside diphosphate becomes the deoxynucleoside diphosphate. And at the very same time, the enzyme, right, gets oxidized. So this molecule is being reduced, and the enzyme itself gets oxidized. You'll see it forms a disulfide bond. Well, if an enzyme gets a disulfide bond, it's useless now, right? I can't use it again. Maybe we've seen this before. Think back, just after glycolysis, we had an enzyme that converted pyruvate into acetyl-CoA, and in the process of doing that, our lipoic acid ended up with, yes, a disulfide bond. And then how did we fix it? Well, we reduced it back to, it opened it, and then we need to oxidize it back to a disulfide bond. Here, we're reducing a molecule instead of oxidizing it, so the enzyme gets oxidized in the process. We form a disulfide bond, and in this case, we need to reduce it back to not being a disulfide bond. It's not a lipoamide, it's actually 2 cysteines that are involved, but we reduce it back, and we do that with a molecule called thioredoxin. So thioredoxin in its reduced form donates some electrons to the enzyme, and the ribonucleotide reductase is once again returned to its active, ready-to-work, reduced form. Well, now our thioredoxin is in an oxidized form. That doesn't help, but I can get a new one. The thioredoxin could be reduced back to its normal form using NADPH. Right, so an NADPH powers this thing, right, along with some O2 and some water and some radicals, which we'll talk about. But ultimately, we reduce our nucleoside into our deoxynucleoside. Okay, so this enzyme has two parts, right? It's two subunits to it. It's an R1 and an R2 subunit. The R1 is the larger of the two. The R2 is the smaller. These are their crystal structures, right? And it has two active sites, right? So if you take one of these, an R1 binds with an R2, so this R1-R2 complex shown down here. Then I have another pair, another dimer of R1-R2 that binds to the first pair. So it's a dimer of dimers. Technically, it's a quaternary structure would have four parts, right? It'd be a tetramer. It'd be an alpha-2, beta-2 tetramer, right? Or R1 twice and R2 twice tetramer. But we call it a dimer of dimers because that's how it assembles. An R1 binds an R2, another R1 binds another R2, and then the pair of that R1-R2s come together and make this dimer of dimer complexes. When it's in this form, it's functional. So it must be in the dimer of dimer or arrangements to be functional. Two R1s and two R2s. Okay? It has three sites that we need to talk about. You remember when we talked about enzymes, we talked about an active site. Right? An active site is where the chemistry happens. Right? It's where all the action takes place. It's where the substrates bind. So an active site here is the little magenta colored circle. It's where the chemistry is going to take place. It's where we're going to do this. We're going to turn this ribose into deoxyribose. That's where it happens at the active site. The specificity site is what programs this machine. Much like our specificity site we saw in proteases, right? The active site of a protease just cut the peptide bond. But the specificity site told it which peptide bond to cut, depending what amino acid would fit there, right? That's how we got chymotrypsin versus elastase versus other serine proteases. So here, the specificity site is where we tell the enzyme what to do. We program it. Okay? Think of the blue circle here as the input site, or the keyboard. We can tell it what to do. The active site is the functional part of it, where it does the chemistry. And then there's this other yellow one with, and I apologize for the name, I didn't come up with it. It's called the activity site. You can think of it as the on-off switch. Okay, so the magenta active site, that's where we do the chemistry. We turn the ribose into the deoxyribose. The specificity site is where we program this thing to tell it which nucleotide to do that to. Okay, not just any one of them, which of the ones should you do. And then the yellow activity site is an on-off switch. If something binds here, this thing works. If something else binds here, it does not work. It's literally an off switch. Okay, so we have three places you can control it. Think of the yellow as a master switch to turn it on or off. Think of the blue as telling it which nucleotide I need to reduce. And the active site is where we reduce the nucleotide from ribose to deoxyribose. Okay, so here's the mechanism, and this is a new slide I put up from the previous ones. I put some more explanations on it. Hopefully this makes more sense. But the way it all starts, we have in the R2 subunit, we have a tyrosine. Right? It's not unusual to have a tyrosine, but we have a particular tyrosine. It's actually tyrosine number 122 in reference to the E. coli one. So all these numbers are in reference to E. coli, because it's the one that's most studied. So in the R2 subunit, the smaller subunit, there's a tyrosine. That tyrosine is a radical, right? So it means it's got, at the end of the benzene ring, there's an OH on a tyrosine. The H has been removed, but we don't have an O minus left behind. We have an O dot, right? It's one unpaired electron. It has no charge overall because we lost a proton and an electron, but the tyrosine is an O dot, right? So it's a radical tyrosine. And that tyrosine is very deep inside the R2 subunit, right? It's virtually inaccessible, right? How did we get the tyrosine to look that way? Well, around the tyrosine, which I'm not going into detail, you can read the paper if you want to look at more detail. I linked the paper on D2L for you. and you at least should read some of it. But inside there's also some iron ions, so the ferric and ferrous ions, there's a pair of them there. And there's also access for an O2 molecule. So at some point, the R2 with its tyrosine and the O2 molecule and some iron ions, it forms a radical, using those ions as the acceptor of the electrons. Okay, so we have an R2 with a radical tyrosine, and it's a rather stable radical tyrosine because of all the things around it. So it's very deep in the pocket there. Nothing can access it except O2, basically. Okay, so when it binds to R1, that tyrosine radical is in no way anywhere near the active site. Right? What it does is transfer its radical electron, right, or absorb a radical electron from neighboring amino acids and iron atoms and lots of other carriers, right, mostly amino acids and iron atoms. But we can transfer one electron from one to the other until we finally are abstracting an electron from cysteine 439, which is in the R1 subunit. And that's where our diagram here begins. So what I'm calling step 0, with a little asterisk next to it, because it's not really part of a cycle, it's how we generate the radical and how we transfer that radical electron or make the radical appear on the cysteine in the R1 subunit. So that's why I'm calling it step 0 here. It only occurs to prepare the system. We only have to do it once. Right? After that, we're good. But we must generate the radical, and we must transfer it to the cysteine. So the tyrosine's radical is made in the R2 subunit by oxygen and iron atoms. And then we transfer that radical onto cysteine 439 in the R1 subunit. Okay? Again, the number is in reference to E. coli, but it's just a cysteine. It's what you need to know. So what happens next is our ribonucleotide, whatever base might be there, binds. The base is sort of irrelevant for the chemistry of this step. We'll talk about the specificity site next. But whatever it might be, the chemistry is the same, whichever one binds, right? And the sulfur here has an unpaired electron. So what it does is it abstracts an electron from the bond between carbon number 3 and its hydrogen. Now, in most of these structures, I'm not drawing that hydrogen, but I threw it here for you because you know there's a proton or hydrogen on here. So between this one radical electron, these half arrows here, and the pair here, one of them forms a new bond to the sulfur and brings the proton along with it, making an SH bond. Okay? That leaves behind an unpaired electron on carbon 3'. So that's shown after step 1 here. We have an unpaired electron on this carbon. So we have a radical carbon, which is not stable. So what happens next is glutamate, as part of the enzyme, deprotonates the OH on the 3' carbon. Right, so we have a deprotonation event here. This abstracts the proton. The pair of electrons in the OH bond migrates to form a carbonyl here, which leaves no room for this extra electron. It migrates over to the 2' carbon. Right? When it migrates to the 2' carbon, then again, there's a hydrogen here as well. It leaves no room for these extra electrons, so the pair of electrons goes on to the oxygen. Right? This oxygen was being stabilized or hydrogen bonded to yet another cysteine, number 225 here, which was protonated, and it abstracts a proton from the cysteine, leaving behind a cysteine with a lone pair, which immediately abstracts a proton from a neighboring cysteine, leaving it with the lone pair. So there's my negative charge over there. These two cysteines might look equivalent, but they're not. One of them has a slightly smaller pKa than the other. I'm going to let you guess which one is which. You should be able to figure it out. If they're sitting next to each other, and this one would rather be deprotonated and this one protonated, you can figure out which one has a slightly larger pKa. Okay? So at this point, when these two electrons left carbon 2 because of the radical migration, we end up with an OH with an extra electron pair, which abstracts a proton from this sulfur, leaving a water molecule, shown in blue. So that water leaves. Right? So we have a loss of water here. Right? That leaves behind a sulfur with a negative charge, which of course abstracts a proton from a neighboring sulfur, leaving it with the negative charge. So that's my scenario at this point. I'm left with a carbon here with a radical. There is one H left on this carbon, but we haven't been drawing it. But we have a carbon here with a radical. Okay? Then we start reversing things. This sulfur, again, attacks the other sulfur. So we're going to form a disulfide bond. Right? When these two attack each other, form a disulfide bond, It does so in a way that it uses only one of its two electrons, not both. Right? So one of these electrons forms part of this bond. You notice the single-headed arrow here, or half arrow, forms a bond here. The SH bond here, one of those electrons participates in forming the disulfide bond that leaves an unpaired electron on the proton. And that unpaired electron then goes and pairs with the other unpaired electron of the carbon, bringing the proton with it to put this H on carbon number 2. Okay? That leaves us with this sulfur still having an unpaired electron and a negative charge, and this proton now attached to carbon number 2. There are now 2 hydrogens of carbon 2. That's what our goal was. Okay? But we need to resolve the situation. We have this carbonyl still on the sugar. That's not good. So our sulfur disulfide bond still has an unpaired electron and a negative charge. That unpaired electron is donated to carbon 3 that disrupts our carbonyl again, and we're kind of reversing our steps. The carbonyl forms an O-, right, with a radical still on the carbon. The O- abstracts a proton from the glutamate, right, or glutamic acid at this point, and leaves behind a glutamate. So we're left with a disulfide bond in the enzyme, right, which we're going to need to fix afterwards. And we're back to having a radical at carbon number 3, much like we were after step 1 up here. So we reverse step 1, right? We protonate that carbon, right? We put the H back on. I didn't draw it here, but this H goes back on carbon number 2 like it was, or sorry, carbon number 3 like it was originally, right? But to do that, I only need one of the electrons in that pair, putting the other of the pair of electrons back on the sulfur at 439, and I'm back where I started. I've made a deoxyribonucleotide now. It's free to go, and a regular old ribonucleotide with its OH on 2 can bond, and we can do this again. I do not need to generate a new radical. I still can use this one I have. I can do this cycle over and over and over until at some point I have a problem. At some point this radical is going to interact with something else, perhaps an O2 molecule, perhaps a water molecule, and we're going to lose our radical. Then we have to go back to the pond, right? Have R2 tyrosine generate yet another radical and give it to a series of amino acids and metals and give it back to the cysteine again. So much like photosystem and photosynthesis, this is acting like photosystem 1. It's donating a radical. In case of photosystem 1, we send a pair of electrons down the chain, eventually one at a time, to photosystem 1 from photosystem 2. Here we're donating it in form of a radical. So very much like that other system where this only needs to donate when we have a problem. Otherwise we're going to keep cycling it, much like we do with cyclic photosystem 1. Okay, so keep that in mind. I'm trying to draw similarities between these pathways. So in order for this enzyme to happen again, a reaction to happen again, of course, we've got to resolve this disulfide problem. And we use our molecule of thioredoxin to do that, which ultimately is reduced by NADPH. Okay, and we can do this whole thing again. So the chemistry of this is much like the setup of photosynthesis, or the light reactions of photosynthesis, and it's much set up like photosystem 1 in its cyclic form, okay, where we only need to occasionally introduce more radical electrons if they get removed. Okay? So that's the chemistry no matter what the base is here, but the really cool thing about this enzyme is how does it know which base to use? Okay? And this is why I call it a programmable enzyme. So we'll spend a lot of time on this slide. This is one of our last slides. And with this slide, I'm going to show you the 3 sites again. So on the very far right, the picture, the really big picture, I want to show you the 3 sites. We have an active site on the right. This is where the chemistry takes place. This is where we turn our NDPs into the deoxy NDPs at this very site on the right called the active site. The 2 sites on the left, the allosteric sites, are either the on-off switch, the activity site, but down below, or the substrate specificity site. This is the programmable site, right? The one above. Right, so it looks like a lot of things are going on, and I actually like this figure because it describes it fairly well. I have 3 things shown on here one thing shown on here 3 different ways for you. Let me do the easy thing first, the on-off switch. So the on-off switch down here, this should make perfect sense from a metabolic point of view. Remind me again, what does this enzyme do? It turns nucleotides into deoxynucleotides. right? It puts the little D in front of them, right? It makes them DNA precursors instead of RNA precursors. So if you have a lot of deoxynucleotides around, you probably don't want to make any more. And if you're lacking in deoxynucleotides, you probably want to make more. So we have an on-off switch that's sensitive to ATP or DATP, right? Of course it's A, right? So if ATP binds, it means it's more abundant than DATP, which means I'm lacking the deoxy forms. So if ATP binds in this pocket, the enzyme is on. It makes more. If I have a lot of deoxy versions, DATP outcompetes the ATP and it shuts the enzyme off. Right? How does it do that? It's not simply an on-off switch. It's not a bulk light switch. How does it actually do this? Remember I said this enzyme works as a dimer of dimers. Right? So shown over here on the left again, on the bottom left is the on state with ATP bound in this site. So green, on, ATP bound there. That means you have it's called alpha, but it's the R1 subunit and the R2 subunit, right? One of each of those binding to each other, and then that dimer forms a dimer of dimers, two pairs of alphas, two pairs of betas, or two R1s and two R2s come together to make this complex on the left, and it is active. So it's an active dimer of alpha-beta dimers. If deoxy ATP binds in that site, that's the red version instead of the green, it prevents the dimerization of dimers. So in other words, it takes the alpha-beta dimer, or the R1-R2 dimer, and another R1-R2 dimer, and doesn't allow them to interact. What it does, it takes 4 of those alpha-beta dimers and arranges them like this, right? So that the active sites can't touch each other, right? So the two have to arrange in a certain way. If you look back over here, that R1 from, looking at the bottom right of the figure here, R2 from, say, one particular subunit dimer gives its radical to the other one's R1, not to its own R1, right? It gives it to the other one's R1. So R2 here at the top gives it to the bottom one's R1. So they contribute to each other's R1. But if you arrange them this way, they're too far apart to do that. That channeling of that radical electron from the tyrosine through the many intermediates down to that cysteine is not possible. So it arranges it in this tetramer of dimers, right? And that's inactive. And that one phosphate difference, or that one OH difference, is what allows that to happen. So it's very sensitive to this. But if ATP is bound, it forms a dimer of dimers, and we can do our chemistry, right? So that takes care of the on-off switch. Very simple on-off switch. So for the substrate specificity site, I'm going to show it to you 3 ways. Let's do it in text first, just a listing up here at the top. So in the specificity site, I tried to color code everything for you here. If ATP or deoxy ATP, so if any form of A, right, binds, this is the orange one up here, binds in the substrate specificity site, or just the specificity site, then the enzyme is programmed to make pyrimidines, right? So if A's bind here, right, in the orange, it will turn pyrimidines into their deoxy forms. So ATP or DATP will form a UDP into DUDP, or a CDP into DCDP, right? It turns the pyrimidines into their deoxy forms, either one. In fact, it'll do both of them, not at the same time, of course, but either one of them could bind here. So if an A form, right, either it's ATP or DATP, same two that bind down here, either one of those binds here, it'll make pyrimidines into their deoxyribose forms. Okay? If deoxy GTP binds here, it'll only turn ADPs into deoxy ADPs. So blue with blue. Right? And if deoxy TTP binds up here, so the yellow one, then it'll make GDPs into their deoxy GDP form. That might sound a little convoluted, right? So the words up here match the figure, but if I tell it to you in a different order, it'll make more sense. Okay? So let's say we have deoxy ADP binding. Okay? Let's just say we have some of that around and it binds in the specificity site. What should this enzyme produce? Well, looking over here, we have deoxy ATP bound, right? It's instructing ribonucleotide reductase to turn pyrimidines into their deoxy forms. So it does. It turns CDPs into deoxy CDPs. It turns UDPs into deoxy UDPs. Of course, those deoxy CDPs become C-triphosphates, right? You know how that happens. We covered it already. And the U's become T's, right? We're going to methylate it, right? So the UDP becomes a deoxy TTP, right? And of course, the UDPs and CDPs, you know, are balanced from the beginning. So the TTP is the only new thing there. So if we build up enough of that, it will displace the ATP that's bound and become the new programmer, right? So the ATP and deoxy ADP will be displaced, and deoxy TTP will take its place in that spot. I've reprogrammed the enzyme now. It doesn't make pyrimidines into their deoxy forms anymore. It turns G's into their deoxy forms. Well, eventually you make enough of those, and the GDPs will turn into GTPs, right? And the GTP will eventually build up and displace the thymine. And so now we're going to move on to the blue part. The GDPs, the deoxy GDPs bind there, and they direct the conversion of ADP into deoxy ADP, right? And of course, once I build up enough DATP, it goes back and says, let's make some more purines. So we have a cyclic nature to this thing. The programming is in a cycle that ensures we get all the right stoichiometric amounts of the deoxy forms, the CTPs, the DUTPs, which is only useful for turning into DTTPs, and then the GTPs and the DATPs. But if we build up enough DATP, right, at first we don't have any, so ATP has to get this started, but if we build up enough DATP, it'll keep cycling through these sets until the amount of DATP far exceeds the amount of ATP, and it binds in the activity site and shuts it off. So it's a really cool programmed enzyme so that you get a cycling through of what to make. It's a factory until you make enough of all the components that you turn it off. All right, so how does it know which one to make? We bind these things here in the allosteric site, and it tells it what to make over here. They're pretty far apart, right? It looks like these two sites are distant to each other. They're actually not that far apart physically. There's enough room for a simple loop of protein in between the two, right? And that's shown in the video I posted on D2L. It's also in the same paper that these slides came from, or that these images came from. It's just also the second paper on D2L. So watch the video first. And it's a very subtle, when these different A's, G's, or T's bind up here, the size of those bases rearrange some amino acids in that really simple loop 2, it's called, and it only allows certain things to bind in the active site. So watch the little video. It's a video representation of a few figures in the paper, and it should explain very simply how this mechanism happens. Okay. And the last thing we haven't talked about, which I mentioned we needed to do, was make thymine. Right? So how do we make thymine? We have a UDP, and the very enzyme we just talked about is turned into deoxy UDP. And then our friendly little nucleotide diphosphate kinase, yes, it works on these too, turns the deoxy UDP into the triphosphate version, just like it would with anything else, the donor being ATP once again. Now this part might seem unusual. We just went from UDP to the deoxy form of UDP. We put a phosphate on it, and then we cut 2 phosphates off. Because our deoxy UTPase can only remove pyrophosphate, right? So we couldn't go directly from UDP to deoxy UMP because my ribonucleotide reductase only works on the diphosphates, and this kinase only works on the diphosphate, so it puts that on. And then my DUTPase will only cut off pyrophosphate, so we're back to DUMP. It's kind of a roundabout way to get here. But it was because of the restrictions on these other enzymes we had to do it this way. So DUMP, right, converts the uracil to a thymine. It adds a methyl group, right? So that's thymidylate synthase. It does this, and it gets the methyl group from the methylene tetrahydrofolate. And then finally, we take the DTMP and fix the phosphate issue and put the 2 phosphates back on by similar enzymes. Our friend here at the end again, right? But this thymidylate synthase is blocked by inhibitors we make like 5-fluorouracil. We talked about 5-fluorouracil once before as an anti-cancer treatment because cancer cells tend to rapidly divide. They're making a lot of DNA, and so they need to convert a lot of U's to T's, right? Much more often than a normal cell. So if we block this conversion of U to T, we will kill the cancer cell. We'll also kill every other rapidly dividing cell in the body. That's why you have hair loss and sloughing off the digestive tract and other things. But it's a suicide inhibitor because if you look at the structure, it looks a lot like a U, like a uracil. Look, compare the two. Here's uracil, and here's 5-fluorouracil. The only difference is this fluorine attached. And that's exactly where we'd like to put the methyl group. The fluorine's in the way. So if we had to try to use this as a substrate, instead of removing a proton here like we would with the uracil, we're trying to remove an F+. F+ is an inherently unstable atom, right? F is the most electronegative atom. You're not going to remove it as F+. So this thing's dead in the water. It's a suicide inhibitor. Once it tries to remove the F, it gets stuck on the enzyme and it never comes off. Right? Go back and look at our articles I put up for you, the Waldrop article where we discuss the different types of reversible inhibitors, and I said to go ahead and read through it. And I said if you want to, go ahead and read the end of it where we talked about an example in medicine. This is that very example. So the 5-fluorouracil acting as a competitive inhibitor, right? Or sorry, a suicide type inhibitor of thymidylate synthase, and we generally give it a combination with something like leucovorin, which locks it onto the enzyme. Okay, so go back and read the end of that if you want this to make more sense along with the enzyme stuff. Right? Where do we get the methyl group to normally do this? We get it from the methylene tetrahydrofolate. And that's this carbon down here in this 5-membered ring is donated to this carbon up here, opening the ring and leaving just tetrahydrofolate. So you need more 5,10-methylene tetrahydrofolate to continue. All right, so how do we continue? We need more of that 5,10 tetrahydrofolate. How do we make that? Well, we converted it to dihydrofolate. Right? And to convert dihydrofolate back to this, we first must reduce it to tetrahydrofolate. I need to put some hydrogens back on these nitrogens here. Right? And then I need to make the ring. But I need a carbon source to make this ring. So my carbon source comes from serine. Right? Serine's going to donate a carbon, right, and leave as glycine. Right? So sodium, or it's not sodium, but serine is donating 1 carbon, leaving as glycine, because that's the difference is 1 carbon. Right? It's the hydroxymethyl it's donating, and that forms this ring. Right? So as long as I keep converting serine to glycine, I can keep converting my U's into T's. Well, this enzyme right here, dihydrofolate reductase, that turns the dihydrofolate to tetrahydrofolate, we've come up with an inhibitor for that, methotrexate. If you look at methotrexate, there's a methyl group in the way. Right? I can't form this molecule by putting a methoxy group on it if there's already a methyl in the way. Additionally, this enzyme over here, the hydroxymethyl transferase, that regenerates the ring, we can't inhibit that enzyme because that would kill all cells, because every cell needs to do this, because it's a source of our methyl donations. But the Plasmodium enzyme and the malaria-causing agent, Plasmodium vivax, its enzyme is slightly different than ours. So if you find an enzyme in an organism that's slightly different from its hosts, that's a great target for a drug. So this enzyme is a target for a lot of anti-malarial drugs.